With that, we move to the next speaker, moving from dark matter projects to algae and bioenergy production plants with Richard Sayre, one of the top specialists in algae and energy production plant research. In fact, Nature magazine is citing him one of the top 5 scientists in the world, one of the top scientists who can actually change the world. And I have nothing more to add to that.
Yeah, thank you. Well, thank you again for the invitation to speak. And I obviously wear a number of hats, and over the last few years been very engaged in the algal biomass community. But really, among all the major issues that are facing the globe currently, perhaps energy is one area that has the broadest implications across the planet. And it's clear from this intersection of economics, domestic security, and environmental issues that energy is going to be a hot topic for some time to come. So this morning I went across the street and looked at the price of fuel at the Chevron gas station. It's $4.37. I live in New Mexico now, and it's about $1 a gallon less expensive. So, but I still drive a hybrid. So, economics has clearly been substantially impacted by the energy economy. For the United States, one of the most important issues, of course, is the issue of domestic security. And the President's policy focusing on sustainable domestic energy production in the United States clearly addresses this particular concern that as much as half of our energy is coming from foreign sources. Many of those countries, of course, which are politically unstable or in turmoil at the present time. And then, of course, the broader impact of global climate change associated with the release of carbon into the atmosphere is a serious concern. So if we look at our current energy economy, oil-based fuels, of course, play a very critical role in the overall picture. A large amount of infrastructure has been developed in the United States centered around oil-based energy economy. It's estimated that there's at least $1 trillion investment in the production, transport, and utilization of fuels that are derived from oil. And the reason that oil is so popular, or such a great fuel source, of course, is that it has, surprisingly, the highest energy density per unit volume of any fuel that's available. That exceeds liquid hydrogen. On a mass basis, of course, liquid hydrogen actually has higher density. But on a volumetric basis, nothing actually exceeds oil other than nuclear energy. And I don't think we'll be powering our cars with nuclear reactors in the near future. And so if we look at the barrel of oil, about half of that energy content in a barrel of oil cannot essentially be replaced by any other substitute at the present time. This is particularly true for aviation fuel. So we can't fly airplanes on batteries. We can't fly airplanes on ethanol because the size of the fuel tanks would have to be twice as big to provide the same amount of energy content. So if we're looking at sustainable alternatives to oil production in the United States, then clearly one of the attractive choices is biofuels. There are a number of clear advantages for producing fuels from biofuels. Most important, of course, is that it potentially is sustainable. It's not an extractive technology, unlike removing oil from geological formations. There is the potential actually to sequester or at least recycle carbon and therefore reduce emissions. And then, of course, many of the biofuel crops, in fact, the algae that I'll be talking about, are actually producing oils as a feedstock. And so that's compatible, again, with the existing infrastructure to produce liquid transportation fuels from petroleum. It's really a drop-in fuel replacement. The major constraint, though, is really what we're dealing with is the very low solar energy density on the surface of the Earth. So in New Mexico and Arizona, which have the highest solar fluxes, it's about 9 kilowatt hours per meter squared per day. Not a lot of energy. There are, of course, other constraints that we have to be concerned about in the production of fuels from biomass. So if we start to take apart the process of solar energy conversion into chemical energy by photosynthesis, we've heard yesterday and a few times during the discourse of this conference that the efficiency of photosynthesis is very, very low. For C3 low efficiency plants, it's about 4%, and it's about 6% for higher efficiency C4 plants such as sugar cane and corn. If we break down where the greatest losses of energy are actually occurring, the largest percentage loss is actually in the conversion of captured energy into chemical energy. About half of the energy loss actually occurs up front in the initial events in photosynthesis, and yet most of the research is really focused on this downstream portion, and that is largely the carbohydrate synthesis, the Calvin cycle, trying to re-engineer ribisco to make it a more efficient enzyme, reduce photorespiration, and so on. And then very importantly, of course, down here at the bottom is the accumulation of that biomass. There's another way of actually looking at this process of photosynthesis, and this is looking at energy efficiency using different colors of light on a kinetic time scale. And so the very early events, the photophysics, are actually super efficient in photosynthetic systems. Light harvesting is 100% energy efficient, and this is at room temperatures. The best analogy to that is superconducting magnets. They're very highly energy efficient, but that's at liquid nitrogen temperatures. And so here we have a system that can transfer energy between pigments with very, very little vibrational energy loss at nearly 100% efficiency at room temperature. And so that has broader implications, of course, in terms of potentially designing optical computer systems that would have to be able to transfer energy at similar efficiencies. But as we get away from the photophysics and down into photochemistry, at slower time scales here, then that's where the inefficiencies start to aggregate. And you can see on this slope here for various different kinds of plants as well as chloroplasts where they fall on this scale. And again, the largest energy losses, and we're using red light because that's most efficiently utilized for driving charge separation, are occurring again in this upper region here. That being said, time to harvest, or which might be called agronomy actually, is critically important for biomass production because if we have a failure in that stage of the process, it may be another year or another season before a crop is actually accumulated. So clearly one of the questions is if we're going to rely on biomass as a source of renewable and sustainable energy production, the question has to be asked, or what are the most promising organisms? And so we are particularly attracted to algae for that very reason. And the bottom line here is if we look at oil yield, it turns out that algae, not only on the oil basis but on a biomass basis, outproduce terrestrial crops by anywhere between 2 and 10-fold on an aerial basis over the course of a year. And you can see the various land areas here that would be required to grow fuel, essentially, using different sources, either corn, in this case, the large yellow box, cellulosics and hemicellulosics, or algae to displace gasoline production in the United States. So if we look at why algae are so good at converting solar energy into biomass, it comes down to a number of important points. And perhaps one of the most important is that every individual cell is actually doing photosynthesis. If you're a photosynthetic microbe, there are no heterotrophic tissues. There is no drag on the system in a sense that you're supporting the growth and respiration of roots and stems. But also algae have more efficient photosynthetic systems. They have carbon concentrating systems. In some cases, it's a C4-like pathway, as in diatoms, but in other organisms, it's an inorganic bicarbonate pump, which is an energy-dependent process. They grow fast, they're facultative in their oil accumulation. These are actually oil droplets stained with a dye inside of a cell. And from a domestic security perspective, one of the attractive features about algae is they can be harvested potentially 24/7 and 365 days a year. And so that reduces risk associated with failures in the system. If there's a failure in a terrestrial crop system in the temperate portion of the world, you're potentially losing energy for a year. But if you have a failure in a pond production system, generally you can be back up in operation in a very short time, within a matter of days. Another very attractive feature about algae is the potential to capture carbon dioxide. And so carbon dioxide emitted from point sources such as power plants or cement kilns or ethanol fermentation facilities can be injected into the pond, hydrated to form bicarbonate. It's no longer a gas, it's now trapped in the algae, pump bicarbonate in the cell, convert it back into CO2, and support photosynthesis. Wastewater utilization from municipal sewage treatment plants, runoff from dairy farms, et cetera, is another source of nutrients. And so nutrient recycling is another feature that's very attractive for utilizing algae as a source of biofuel production. And then no direct competition for food. Now, it's arguable that the land area that would be utilized for producing algae could impinge upon agricultural land areas. But I think it's important to point out that the delipidated biomass, the remaining protein and carbohydrate, after the extraction of the oil from the biomass, is a very high-value product and can be used for animal feed. In fact, the estimates are that if we use algae to produce all the liquid transportation fuels in the US, the amount of protein that would be produced as a co-product would displace all the protein currently produced by corn and soybeans. And so it's an added value product and not actually a direct competition. So we have a number of projects ongoing in the lab actually trying to address increasing the efficiency of biomass production utilizing algae, and they range from projects that address very, very fast events in the microsecond and shorter time intervals to things that last for days or so. The story that I'll be telling you about today is really trying to optimize energy conversion efficiency. And as I pointed out earlier, this is an area that's generally received not a lot of attention, even though a large fraction of the energy in efficiency in photosynthesis actually is associated with the capture and conversion process. So there are a number of people that were involved in the project, and particularly Zoe Perrine and Sangeeta Negi's work is what I'll be talking about today. And very importantly is our collaborator, Jerry Timlin at Sandia National Labs. And so the picture that you're seeing here is actually shown in this cartoon over on the right here. And what you're seeing is the association of the light harvesting antenna complexes with a dimeric photosystem II reaction center complex. And so you can see these trimeric LHC complexes associated with these reaction centers where the photochemistry actually takes place. And to sort of skip ahead for part of the story, it turns out in plants and in green algae, which have a photosynthetic apparatus that are virtually identical in many respects, these antennas actually can move. And they can move laterally in the membrane, and they can redistribute energy between the different types of photosystems. So if we look at the process of photosynthesis in all plants and in all algae, it all light saturates at 1/4 of full sunlight intensity. That's a big deal, that it light saturates at 1/4 full sunlight. In New Mexico, they keep saying we're a great place to grow algae because we have all this sun. Well, the bottom line is it's no better there than it is in Ohio, where I used to live. And in Ohio, they have this gray linoleum sky all the time, for those of you that don't know that. But anyway, so photosynthesis, light saturates at 1/4 full sunlight intensity. But the pigments, of course, are still capturing photons. That's physics. That's something that we can't really change. But what do they do with those excited states? Well, what they do with those excess excited states is they dissipate that as fluorescence or as heat. And that process is generally known as non-photochemical quenching. And so what we'd ideally like to do to increase the efficiency of photosynthesis, of course, is convert all of those excited states into charge-separated states that generate reduced carbon molecules or carbon-carbon bonds in the sense of biomass. So to understand how to manipulate this process, we have to go through a cartoon here. And this cartoon shows the photosynthetic electron transfer chain in green plants and also in the chlorophyte version of the green algae. There are 2 photosynthetic reaction center complexes, photosystem 2, which of course splits water. This is the source of the Earth's atmospheric oxygen. I like to say it's the most important biochemical reaction on Earth, which maybe is heretical in this audience. Everyone likes DNA. But we're all supported by this because this is where the energy that's available for life really starts. So oxygen is generated here. Electrons are transferred by mobile electron carriers along with protons. And actually, this coupling of electron and proton transfer is one of the rate-limiting processes in photosynthesis. It's a subject that we're addressing from other strategies. But here at the cytochrome B6F complex, the protons are dumped into the lumen. And of course, those protons and the protons from water splitting drive ATP synthesis. The other photosynthetic reaction center complex is photosystem 1, and it reduces by linear electron flow NADP, and of course NADPH and ATP drive the Calvin cycle. Now, what's shown in these yellow boxes here are the rate-limiting steps for each of these processes. So the light harvesting apparatus, as I mentioned earlier, is super efficient, and those kinetics are very, very fast. Photosystem 1, the rate-limiting step is 1 microsecond. The overall rate-limiting step here is the oxidation of plastohydroquinone. That's a millisecond. And photosystem 1 is actually faster than photosystem 2. And as a result, it actually doesn't emit much chlorophyll fluorescence because there's very fast electron transfer here. So we've got very fast kinetics, many orders of magnitude faster than this process. And so it's clear that if we're putting in photons at a very high flux rate, that we have a kinetic constraint here. And it's really these kinetic constraints that result in the dissipation of a lot of that energy that's captured as heat or fluorescence. So about 10 years ago, or well, now it's 20, I guess. Wow, that's a long time ago. Mellis's group at Berkeley, and in particular Jürgen Pohle, showed that if you eliminated the light harvesting complex, and they achieved that by finding a mutant that no longer made chlorophyll B. Chlorophyll B is only associated with the antenna. It's not part of the reaction center. But they found that these organisms with these small antennas actually did not light saturate at full sunlight, or at 1/4 of full sunlight intensity, and had higher rates of photosynthesis. And so this was the impetus for trying to actually make this system even better. And so that's the story that I'm going to be telling about how we actually improved upon this initial observation. Okay, so effectively what they were doing by eliminating the LHC complex was reducing the optical cross-section that actually intercepts or absorbs the photons from the sun. But it turns out that the LHC has many functions, largely associated with the light harvesting apparatus. But one of those functions is that, as I mentioned earlier, is that it can actually migrate from photosystem 2 over here to photosystem 1 and redistribute energy between the 2 photosystems. And so that serves the function of balancing overall linear electron flow so that the 2 photosystems are turning over at similar rates. So at high light intensities, the antenna is actually larger with photosystem 2 than photosystem 1. It soon saturates electron transfer, and so the LHC migrates over to photosystem 1 and shares energy there. There's another important process associated with LHC complexes, and by the way, it's a very large gene family with different members that have different functions. And that is, there is a cycle of conversion of carotenoids from a form that actually absorbs light more or less in the green region of the spectrum and serves as an antenna function, providing extra energy to chlorophyll, to a form that's shifted in energy levels that actually dissipates excited states from chlorophyll. And so it actually quenches extra energy when there's too much energy in the system. So if we have excess excited states, and what happens is that there's overreduction of the quinone that leads to an increase in the proton concentration in the lumen, and that activates these enzymes called violozanthine deepoxidases that convert violozanthine in a series of deepoxidase reactions into zeaxanthin. And then zeaxanthin dissipates the excess energy very efficiently as heat. So this process of actually migrating the LHC complexes between photosystem 2 and photosystem 1 also has another very important function. And this is particularly important in algae. And that is, by providing extra energy to photosystem 1, it can support very high rates of what's called cyclic electron transfer. And cyclic electron transfer, in contrast to linear electron transfer, drives the movement of protons across the membrane to support the synthesis of additional ATP without leading to the reduction of NADP. So it's a way of balancing ATP and NADPH requirements for the cell for different purposes. Well, it turns out that in algae, which have these bicarbonate transporter systems, they require ATP for the active transfer of bicarbonate. And so the ability to make ATP, extra ATP, by cyclic photophosphorylation is critical for the overall photosynthetic efficiency of algae. And so eliminating that process or reducing it could have substantial impacts on the overall rate of photosynthesis. So to skip ahead here, what I'll be talking about is actually what are the implications of altering the ability to transfer energy between the photosystems by state transitions and the ability to carry out these dissipations of excess excited states by the violozanthine-zeaxanthin cycle in algae that have various different sized antennas. So the question we're going to ask is, what is the optimal antenna size for algal growth? What are the secondary effects of altering the antenna size? And do higher photosynthetic rates actually correspond to higher growth rates? So the hypothesis is that by reducing chlorophyll B levels, we'll decrease the size of the antenna, we'll reduce self-shading, we'll reduce non-photochemical quenching or energy losses via this violozanthine-zeaxanthin cycle, and will improve the overall efficiency of energy conversion. And so there's a very simple, straightaway approach to trying to alter the antenna size, and that's actually regulating the activity of this enzyme that converts chlorophyll A into chlorophyll B, called chlorophyll A oxygenase. And so the approach was essentially to generate a family of transgenics with different sized antennae using RNAi strategies. So the easiest way to actually screen for these antenna size mutants is looking at the chlorophyll A-B ratio. So if we're changing the denominator, in other words, reducing B content, then the ratio goes up. So what I'll be focusing on from now on is wild type, a couple of intermediate sized antennas, and then of course the mutant which has no antenna. So first of all, if we look at the antenna complex, which is shown here on a non-denaturing gel, here's the wild type level. This is the chlorophyll B-less mutant, again confirming it has no antenna. These are the intermediates, and clearly there's a reduction here. This can also be imaged by chlorophyll fluorescence under steady state illumination. One way of actually very quickly measuring the size of the antenna is to measure chlorophyll fluorescence rise kinetics. So if the antenna is large, which is the black line here, and the wild type, the rise kinetics are faster. More light is being absorbed, and the photosystem II complexes get over-reduced and dissipate the excess energy as fluorescence. The chlorophyll B-less mutant is the lighter green, and you see its rise kinetics are slower. And here's a family of intermediates here that are somewhere in between these two extremes. But the nice story here is we get a very tight linear relationship between chlorophyll AB ratios and the rise kinetics. One of the questions, though, you need to ask is if you're starting to alter the size of the antenna, does this have indirect implications on the number of reaction centers? And so are we losing reaction centers if we get rid of antenna? That's not a good thing. We wouldn't want that to happen. And so one of the cool technologies down the road in Albuquerque is a hyperspectral imaging technology that's very high resolution. We can actually image complexes in cells, and very high spectral resolution as well. And this is Jerry Timlin's group. So the good news from that analysis is that as we alter the LHC content here, we're not really seeing any substantial change in the number of photosynthetic units. And so the capability to do photosynthesis is not impaired. But what about the impact on those other processes that the LHC complex carries out? So if we look at the viola-xanthan-zeaxanthin cycle, and here what we're looking at is in wild-type organisms, the change in the abundance of zeaxanthin under high light versus low light conditions, we see about a threefold change, which is what's normally expected. So it's protecting the system against excess excitation. But in the intermediates, it's actually working even better. And this was a surprise. This was not expected. So in the intermediates, we're seeing that the ratio here from low light to high light increases by six-fold roughly. And so that's telling us that we haven't impaired that particular function. The other question is, are we altering the so-called state transitions or the migration of LHC between the photosystems? And the answer to that is no. It works very, very well in the intermediates. But in the mutant, which has no LHC content, they can't do this. And this is best represented here by looking at chlorophyll fluorescence rise kinetics. And when we give photosystem I light, what happens is it drives the movement of the LHC complex back to photosystem II, and you get higher fluorescence increases, which is an indication of this particular movement of the LHC complex. We don't see that in the mutant, and so that is impaired. So what happens to photosynthesis? Well, the really great story here is we've increased the rate of photosynthesis at high light intensities by more than twofold. That's a substantial increase in the rate of photosynthesis with these intermediate-sized antennas. In fact, they perform better than the original mutant that was isolated in the Mellis group, which has no peripheral antenna complexes. If we look at growth rates, and here it's very important to note we're looking at growth rates in a small Erlenmeyer flask. And in this environment, not all the light is going to be absorbed. The optical density of the pigments is not sufficient to absorb all the light. That's going to be important, as I'll come back to later. But under low light conditions, the intermediates and the wild type are very similar. But under high light conditions, we're getting a 30% increase in biomass productivity. So it translates over. So the question is, in a cultivation system, how do you take advantage of a monoculture of organisms that have smaller antenna sizes? And so the hypothesis here is that with wild-type algae, with these very large antennae, they're dissipating a lot of the extra energy they're absorbing as heat or fluorescence. Plants and algae are light hogs. I'll come back to that in a moment. But with small antennae, of course, they're intercepting less light, so light will penetrate deeper. So the prediction would be is that with small antennae, they'll do better at producing biomass. They're more efficient in deeper ponds. So to test that hypothesis, we did a field trial in a greenhouse, and we had different depth ponds as indicated here. And we took the large antenna and the very small, and then we did a mixture of the two, as shown here in the middle. And we ran this for 6 months, from February until June. We harvested the biomass every week and measured the amount of productivity. And I'll show you those results in a moment. But there is one important point that I want to make here. What we're looking at in the histograms is over the course of the growth over a week is the light penetration, the photosynthetic active radiation in that pond. And what you can see there is with the small antenna, the yellow, so they're all starting at about the same point, but within one day, we're already starting to get a segregation going on here. And with the mixture of large antenna and small antenna, what you see here is that the small antenna and the mixture virtually look identical over time, which is telling us that in that mixture of cells with large antenna and small antenna, that the large antenna guys are winning. They're outperforming the small antenna organisms. And I'll come back to what that means in a moment. Okay, so if we total up the biomass over that 6-month period of time, roughly 4 months, I guess, and look what's happening, then what you see is that as predicted, the large antenna size will do well in the shallower ponds, but the small antenna does very well in the deeper ponds and actually is the overall winner. So it's clear that by optimizing the antenna size and then building in a production system that takes advantage and recognizes that increase in efficiency, that we can get increased biomass. But this is with a static system. We generated an intermediate-sized antenna, which has very little room to move around. Of course, the amount of sunlight changes annually a great deal, and so ideally what we'd like to do is have an antenna that can optimize itself throughout the course of a year. So the strategy that we took to try to get a self-regulating antenna was the following. We started with the mutant that makes no chlorophyll B, and then we complemented it with a gene construct that would allow it to make chlorophyll B. But on that gene construct at the 5' end, we included a light responsive element to which a transacting factor called NAB1 binds. And when NAB1 binds to this element, it prevents the translation of that messenger RNA. And so NAB1 levels are very light dependent. So under high light conditions, there's a lot of NAB1. It'll suppress the translation of this transcript, and we'll get small antenna size under highlight, which is what we're shooting for. Under low light, it's not expressed, the NAB1 protein, or at very low levels. And so under those conditions, we'll get highlight. And so does it work? And the answer is it works very nicely. And so here is the change in the antenna size from large to small. And we're measuring chlorophyll fluorescence rise kinetics as our surrogate for antenna size. And under control algae, you see there's no clear pattern. They're not very rapidly adjusting the size of the antenna. If we mutate the light responsive element so that it no longer interacts with the NAB1, the same story holds up. But in the wild type NAB1 construct, we see ideally what we had hoped to achieve. So under low light to high light shift, the antenna gets smaller. Under high light to low light shift, the antenna gets larger. And these are just independent transgenics. There's always, of course, the one guy who doesn't behave. But anyway, what we're seeing is change in the chlorophyll AB ratio from 2.4 to 3.6, right in that sweet spot for photosynthesis. So to summarize, really, I think what are the important take-home messages here is that in our original hypothesis, we got 3 of the 4 correlates right. One of them was wrong. We did not anticipate that the violozanthan-zeaxanthin cycle in these intermediates would actually be better, and that turned out to be the case. But there's really a very important take-home message, and that is that algae and plants are really light hogs. They capture energy, and they do this because you might think of it as a limiting nutrient in the environment. And if you're better at capturing it than anyone else, even though you're dissipating that excess energy as heat or fluorescence, you win. And so it's non-intuitive that photosynthesis, by being inefficient, actually provides a selective advantage. It's not something that you would have expected. But the implications for agriculture are also critically important as well. Under monoculture conditions that are highly managed, and so we can eliminate the competition from the light hogs, how would we design a crop plant to be more efficient at photosynthesis? And one strategy is that we would engineer so that the upper canopy leaves would have smaller antennae, so they're more efficient at utilizing those high-intensity fluxes of photons, and the lower leaves would have the larger antenna. And so that's a project that's currently ongoing, and we're hopeful that we'll be able to improve the overall photosynthesis. So there are many people involved in the project. This is the current team, and I'll point out that Sangeeta was very much involved in the analysis of the carotenoids in the project. And I also like to acknowledge the support of the Department of Energy. Thank you.
Thank you, Richard. Questions?
Very interesting stuff. In the marine realm, there's two observations that have been made, and I just wondered, with your expertise, if you could comment on them. The first one is viral PSBA genes, right at the heart of PS2.
Yeah.
They often have mutations in the pest domain. So I'd love to hear your thoughts on that.
Yeah.
And the second aspect is probably the dominant nitrogen-fixing cyanobacterium in the ocean lacks PS2. And I'd love to hear your thoughts on that.
Lacks PS2. So it, yeah. Huh. So I'm not familiar with that story. So it's a chemoautotrope, or?
Well, all that's known is that it has PS1,
Cyclic, yeah. So it's got to be able to, of course, get electrons to carry out carbon reduction from some source. So it's either going to be reduced carbon in the environment, or it's going to be reduced metals in the environment. Those are the sort of the two choices that we know from other photosynthetic organisms. Yeah, I guess that's all I can say. It's got two other choices besides using light to drive electron transfer. And then, in terms of photosystem genes, for example, PSBA. So for those of you that don't know, that encodes a protein in the reaction center called the D1 protein. What's interesting that we know from comparative studies of D1 protein sequences, and Ken can actually address a lot of this, he's working on this now actually, is the fact that if we compare the protein sequences of cyanobacteria to higher plants, one of the very interesting features that's been identified, and this was actually this story was in part developed in our laboratory, is that we know that the quantum efficiency of charge separation in cyanobacteria is about 20% less than it is in higher plants. And that can be very easily measured. And we actually know the molecular reason for that. And the reason for that is that the hydrogen bonding interactions of the protein with the primary electron donor called pheophytin. And in higher plants, there's a very strong hydrogen bond provided by a glutamate. This is a glutamate that's buried in the middle of the membrane, an environment where you wouldn't expect to see a very charged residue. And in the cyanobacteria, it's a glutamine, a less strong hydrogen bonding residue. So we've altered these amino acids at that particular site from very strong hydrogen bonding interactions to very weak. And what we find is that there's a very tight correlation between the hydrogen bond strength and the redox potential of that electron acceptor. And by providing a very strong hydrogen bond, you make it a much more oxidizing midpoint potential. It serves as a better electron donor, and so using Marcus theory, you actually get a better electron transfer. So, so there's a range of efficiencies in photochemistry that can be actually associated with single amino acid changes, and so that may be in part explaining why you're seeing such diversity as differences in photosynthetic efficiencies. Yeah.
We don't have any more time for more questions. I'd like to thank Richard again.